Okay, done. So we are here. So now you know what is amino acid, right?
Mm-hmm.
Any confusion in it? Pretty much simple, right? Okay.
Yeah.
So R is there are 20 R, like 20 R. There are 20 different types of R. Okay,
Mm-hmm.
good. And now I will tell you the 20 names of the 20 amino acids. Okay,
Mm-hmm.
I have a very good friend whose name is Alvi, Alvi, right?
Mm-hmm.
Okay, so I say him Alvi. So A is for alanine. Okay,
Mm-hmm.
L is for leucine.
Mm-hmm.
V is for valine.
Mm-hmm.
ALVI for isoleucine.
Got it.
Okay.
Mm-hmm.
And I, with respect, I say him G, LVG, LVG. So G is for glycine. Okay.
Got
These are
it.
called aliphatic amino acids. These are called
The amino acids.
aliphatic amino acids. So
Alpha.
LVG, LVG,
LV.
G, LVG, right?
Mm-hmm.
Okay. Then we have acidic amino acid. Acidic amino acid. So I
Mm-hmm.
say it AG, aspartic acid and glutamic acid. Aspartic acid and glutamic acid. You can see here.
Oh, I see.
Aspartic acid and glutamic acid. They are acidic amino acids. Okay.
Mm-hmm.
Then I have basic amino acid. I call it HAL, HAL. Right.
Mm-hmm.
This is here histidine, arginine, lysine. Right.
Mm-hmm.
Then there are aromatic amino acids. What?
Aromatic.
Aromatic.
Amino.
Aromatic. 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 Aromatic word derives from aroma,
Mm-hmm.
and aroma is a beautiful thing, right?
Yes.
I am feeling the aroma of the food, right? So aroma, aromatic amino acids. See?
Mm-hmm.
Okay. Take T and P. Take T and P. Tyrosine, take T. Tyrosine, tryptophan, and phenylalanine. You can see here, this is phenylalanine, right? This is tryptophan, and this is tyrosine.
Mm-hmm.
So I say take T and P. T, T, P.
Got it.
All right? TTP, TTP.
TTP.
Okay?
Mm-hmm.
So then we have uh, neutral amino acids, which are GATs, GATS, GATS. Okay?
Mm-hmm.
Glutamine, asparagine, threonine, serine. Glutamine, asparagine, threonine, and serine. Okay?
Mm-hmm.
And then we have sulfur containing amino acids.
What was that one?
Sulfur-containing amino acids. Okay, so I went to the doctor and he diagnosed with me sulfur deficiency, right? And then he told me, I said, okay, what is the solution? He says, okay, eat McDonald's, MC, okay,
Mm-hmm.
methionine and cysteine, okay.
Mm-hmm.
Now count how much did you count? How much you wrote on your notebook? Like count, just I mean, tell me with counting, like rate with counting.
19. Maybe I counted that wrong. 18.
18. You count again. Just like I mean, name them for me.
So for
Alanine,
the
alanine,
alanine, leucine, valine, isoleucine, and
I,
glycine.
okay,
And then for the acidic amino acids,
aspartic acid, glutamic acid.
aspartic acid and glutamic acid.
Mm-hmm.
And then basic amino acids are histidine, arginine, and lysine.
Okay.
Aromatic amino acids are tyrosine,
Take
trypto,
tryptophan,
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 and then
phenylalanine.
phenylalanine.
Yeah.
And then for the neutral amino acids, there's histamine,
Glutamine,
glutamine. Oh, oops, that was no. Glutamine, asparagine,
asparagine,
and then
asparagine,
asparagine,
and
and then triodine, and then serine.
threonine and serine. Yeah.
And then sulfur containing is
Methionine
methionine, and
and
then cysteine.
serine.
Methionine.
Cysteine, sorry, cysteine. Yeah. It should be 19.
19, 2, 6. 19. Yep, I counted 19 again.
Yeah, and the last one is cyclic amino acid. Cyclic.
Cyclic amino.
That is proline, right? Praline is a ice cream I like most. So proline.
Oh, I see.
Proline. Got it?
Yes.
Okay. In our lab, we are not usually concerned. These are called 20 proteinogenic amino acids. 20 proteinogenic. What does it mean? Proteinogenic.
Genic.
Means genesis. Whenever comes genesis, the word genesis means something that
Beginning.
makes thing, right?
Mm-hmm.
So these are the protein-making amino acids.
Got it.
Right?
Mm-hmm.
Proteinogenic. They are also called standard amino acids.
Mm-hmm.
They are also called
Standard amino
acids. Why are they called standard amino acids? Because in this particular case, the amino group and carboxylic group will always be attached to central carbon. Okay? Anything can be called amino acid if they have amino group and carboxylic acid group.
Got it.
But if they are not connected with the common C, if they are not connected to the common C, then we cannot say that they are standard amino acids or the proteinogenic amino acids. We will call them unnatural amino acids.
Unnatural?
Unnatural or non-standard. Very boring. Huh? Is it boring?
I think it's interesting. I never learned any of this stuff in high school, so
okay.
I think it's interesting.
Okay. Yeah. So
yeah.
These are the 20 amino acids that make all the proteins that are present in our body, right?
Mm-hmm.
And now there are three ways. Scientists are very, clumsy people. They are very difficult people, right? But I try to make science easy. So there are two ways of naming them. One is 3-letter code and other is 1-letter code. Okay?
Mm-hmm.
Either we write the complete name, or we write the 3-letter code, or we write just 1-letter code.
I see.
Okay. And in peptide chemistry or in peptide, we usually write single letter code. Got it? And mostly, so I will start with the way you have written, right? You have written in your notebook. So I will say alanine is ALA or A, right? Leucine is LEU or L. Valine is VAL or V. And isoleucine is ILE or I. And G is glycine, GLY or G. Right? Yes. Then we talked about the acidic amino acid. This is different now. Aspartic acid is ASP. But it is single letter, it becomes D. Okay? And glutamic acid is GLU, but in single letter, it becomes E. So you just note this because this is different, right? Just circle this, D and E. Right? Because usually other are starting like with leucine, L, isoleucine, I, alanine, A, glycine, G, valine, V, right? So then I told you about the acidic amino acids, right? So in acidic amino acid, we have histidine, which is H. And arginine, this is different now. Arginine, we will say R. Okay? And with glycine, we will say K. Got it? Then we have polar amino acid or neutral amino acids, which is glutamine. So note that glutamic acid and glutamine, right? So there is little different. We will go into detail in it. It is GLN or Q.
Got it.
And asparagine is N. Then I talked about the tyrosine, tryptophan, and phenylalanine. So tyrosine is Y. TYRY. And phenylalanine is F. While tryptophan is W.
Got it.
Okay. Then we have sulfur-containing amino acids, which is M, methionine, or methionine, and then cysteine is C.
Got it.
Then we have what? Proline is P. In our lab, right? Or in the cell penetrating lab. So wherever we are in the cell penetrating peptides, we are concerned about the cell penetrating peptides. So mostly we are concerned with H. H is what? Histidine, right? R, which is arginine. Then we talk about lysine, which is K. Then we tryptophan, which is W. Then we have phenylalanine, which is F. Aspartic acid D. Glutamic acid E. So mostly we are concerned with these amino acids. Mostly, not all the times, but mostly H, R. So if I say, Sarah, give me W. So what is that?
Tryptophan.
Yeah. If I say you give me R.
Arginine.
If I say give me K.
Lysine.
Oh. If I say you give me D.
D.
Aspartic.
Glutamic.
Aspartic.
Oh, aspartic acid.
I say you give me E.
E is going to be glutamic acid.
So it's not going to consume your much time. Just spend kind of 15 to 20 minutes on this table, right? And and also I want you to look at this is R, which is like yellow, right? This is like which is kind of which color it is, like light orange or what, right? So so these are different,
Right.
okay? And when I say charge, it's positive charge, which is acidic amino acid, right? So why it is acidic amino acid? We say that this is NH2,
Mhm.
this is COOH.
Mhm.
They get neutralized. But then we have extra NH2
Mhm.
that makes them positive.
Got it.
Then we have here, and see here, we have NH2 here, we have COO minus here,
Mhm.
okay, neutralized, but then we have two NH2 group here,
Got it.
which makes them positive.
Mhm.
Then we have here NH2 plus, right? This is neutralizing, but this is here, right? So
Mhm.
that makes them positive. And if we see here in negative, we will have extra carboxylic acid.
Got it.
So we have this standard structure, but then we have carboxylic acid group here. Then
Mhm.
we have carboxylic group here, right? And then
Mhm.
we neutral, when we talk about neutral, so you see that this is COOH, NH2, neutralized, overall no charge,
Got it.
overall no charge, overall no charge, overall no charge, right?
Okay. I see.
So that's why we say that, okay?
Mhm.
And in aromatic amino acid, we see this benzene ring,
Right.
this benzene ring,
Yeah.
okay?
Mhm.
So this is how you have to look into the structures. Okay, and now comes the different, like difficult science, little difficult science. If you are not clear about it, don't jump into peptide chemistry. Okay, because this is the backbone of the peptide. Now I asked you what is peptide. Okay, in the start of my session, what is peptide? So peptide is
Peptide is the protein.
no, no. Peptide and protein are different things, right? So when two or more than two amino acids are condensed, you write this. When two or more than two amino acids are condensed,
Mhm.
okay, by the removal of water, they make peptide. Okay,
Mhm.
that is usually between 3 to 40 amino acids. Okay, so
Mhm.
if you have like around 40 amino acid structure, right, that is peptide. But if you have like more than 40 amino acid structure, that will become protein.
Oh, I see.
Got it? For
Mhm.
example, we have insulin, we have hemoglobin, we have myoglobin, thousands and thousands of proteins. So peptides have usually less than 40 amino acids. I'm saying usually, not all the times. Please
Mhm.
be careful in the words, right?
Mhm.
So what is peptide?
Peptides are two plus amino acids, or two or more amino acids condensed by the removal of water, which usually.
Now is a big term here, condensed, right? Condensation, right? The word condensation, write it, make it a heading. Condensation, joining of two molecules, two smaller molecules, joining of two smaller molecules by the removal of water is called condensation. Okay, what have you learned so far?
So far, I've learned how to group the amino acids. based on their R groups.
Mm-hmm.
And then I learned that peptides and proteins are not the same,
Mm-hmm.
and that condensation is a very important process of making peptides.
Making everything in biology. Okay.
Mhm.
Whenever two smaller things combine, right? And by the removal of water, that is condensation. When it's a big term, I'm saying it's a big thing, right? This is basic science. Most of the people don't understand it and appreciate it. Like the bigger level, we are talking about a bigger level. On a bigger level, wherever you go, wherever you go. I mean, this is actually my lecture that I used to give people. So you see this glucose and glucose, right? They are removing water and becoming maltose.
Mm.
Okay, glucose and fructose. Just see it. Don't write it. I will give you this handout. Glucose and fructose, removing water, becoming sucrose. Galactose, removing water, becoming lactose. Amino acid and amino acid, removing water, becoming dipeptide. So majority of biological molecules are polymers formed by condensation reaction, right? So this is very important. Reaction involving joining of smaller molecules by the removal of water is called condensation. Okay. So if it is like, if I add one more here, one more glucose here, like I add maltose with one more glucose, right? It will become like it will have three things in it. So if I talk about three amino acids, that will become tripeptide, right? If I add 10 amino acids, it will become decapeptide,
Mhm.
right? If I add 9 amino acids by this process, that will become nonapeptide. Okay, there are two very popular nonapeptides in our body. Okay, which is called oxytocin. Oxytocin, very powerful thing. Very powerful thing. Oxytocin. All the drama happening in the world is because of oxytocin. Oxytocin.
Mhm.
It is nonapeptide. It is
Nona.
nona,
Nona.
nona, nona,
Nona
nona,
peptide.
nona, nona, nonapeptide. Okay, nine
Mhm.
peptides, 9 amino acids. Nonapeptide means
9 amino acids.
okay, and another is vasopressin. VASOPRESSIN. Yeah, SSIN. Vasopressin, or also called, it is also called antidiuretic hormone.
Ah.
It is also what? Nonapeptide.
But
Both are produced from the posterior pituitary. Both are produced from the posterior.
I missed that last part.
Yeah, so the both oxytocin and vasopressin, they are produced from the posterior pituitary gland.
Oh, in your brain.
Yeah.
Mhm.
Not in brain. It's just behind our eyes. It's just below our eyes. If you go from here down, if you
Mhm.
go from here little down, down.
Right
So
behind.
our brain is like this. Our brain is like this.
Mhm.
Sorry, yeah, this, right? So it's
Mhm.
here, right here, right? We can see that, but it's okay. So oxytocin is a very dangerous thing. It's called cuddling hormone. It is called cuddling hormone, right? It is produced in mothers a lot. That is why they hug their kids a lot, right? Right. So kind of this is kind of cuddling hormone. When you cuddle someone, right? That is like a lot of oxytocin is coming out of your body, right?
Mhm.
And interestingly, you can make this cuddling hormone in your lab. Not difficult process. Just take the amino acids and then I will show you oxytocin structure. Oxytocin, oxytocin amino acid sequence. So see this CYIQNCPLG. Now you have to tell me what is C.
Cysteine.
Very good. What is Y?
I can't remember why.
Tyrosine.
Tyrosine.
Tyrosine. Then I is
Isoleucine.
isoleucine. Q is
Don't remember Q. Um,
yeah. No, go to the notes and check it.
mm, oh, was it triiodine?
No,
Oh
no,
wait,
no,
no,
no.
oh,
Glutamine.
glutamine. Glutamine.
Glutamine. N is aspargine. N is aspargine.
Aspargine.
C is cysteine.
Cysteine.
P
P
is proline. And L is leucine and G is glycine. Right? So if you just connect them, in our lab, we have all these amino acids. If you just do we can get vasopressin. This is vasopressin. Sorry, this is oxytocin. This is oxytocin. But then we have vasopressin as well, right? So again, this is a nonopeptide. So this is a nonopeptide because this has 9 amino acids. So that was the reason. If you are unable to know you have to just. It should be in your second memory. Like what is Y, what is Q, what is N, what is D, what is E, what is L, right? So you should know this. It's very important. Okay. Clear or not clear?
Yeah, clear.
What is clear?
We need to know.
Well, so if we have vasopressin, so what is the difference between? This is a question for you. What is the difference in structure of the vasopressin and oxytocin?
It has the tyrosine and then phenylalanine. Whereas the oxytocin has tyrosine and isoleucine, so that already makes it different.
I don't know. I'm not able to know. Yeah. So you have this. So we have this here, okay, and we have this here. You just have to see what is the difference between both. Where it is, like if you just change and their structure, like little change in the structure makes very different hormone. ADH has all to do with our urine, right? Urine production and volume of the urine control, right? While oxytocin has all to do with emotional reactions, right? So, and there are thousands of peptides in our body. Okay, so now you know what is the difference between peptide and protein, and I just gave you a real example, live real example of two nonapeptides. Okay, so interestingly, now the science has so much developed that you can come to our lab and start making vasopressin and start making oxytocin, right? So is it not interesting?
I think it's interesting.
Yeah, it's interesting that you can make the hormones in our lab. Okay, and there are many. So basic thing. I just want to know what is condensation. You should know that. So basically, peptide synthesis. Peptide synthesis is a condensation reaction. Peptide synthesis is a
Condensation reaction.
and hydrolysis is opposite of condensation. You can see here breakdown of larger molecules by the addition of water into smaller molecules. You see? So you just reverse this thing here. Sucrose, add water, it will become fructose and glucose. Dipeptide, add water, it will become amino acid and amino acid. Got it? So, for example, digestion is an example of hydrolysis. Right? So we take protein, and in our body, like in our stomach, so pepsin, hydrolytic enzyme, it digests the protein into amino acids. First into peptides and then into amino acids, right? So you got it. And then these are the few important functional groups that you need to know. For example, aldehyde, I told you carboxylic acid, ketone, amino group, alcohol, acetylamide, and then important bonds or linkages, So this is like if we connect with this is carboxylic acid group with alcohol group, okay, that becomes this bond, and this bond is called ester bond,
Mhm.
right? And okay, I don't want to go into the details. This is biochemistry, pure biochemistry that I teach. Anyways, any questions so far?
Um, I think I'm okay for now.
Oh, yeah, good. So in our lab, what we do?
We do peptide synthesis.
We do peptides. We make peptides, right?
Mhm.
And I will explain you in our next session maybe what is peptide synthesis, how it is made. But yeah, we cannot explain you if you don't know these basic concepts. These are basic concepts that are taught in high school, but unfortunately they are not taught this way, right? But
Yeah.
it's okay. So I just want to say a good scientist is the one who has good high school.
Mhm.
Okay, if one has a good high school means good high school, right? Connecting dots. Why I am studying? Why I am studying? Why should I study this? If I know the answer to why, everyone will learn to study. Everyone will jump to study. But most of the times we don't know why I am studying algorithms, why I am studying Pythagoras theorem, why I am studying this or that, right? So it becomes boring. Now is another boring thing that I have to teach you, and this is very boring thing. Scholar dot scholar dot google dot com. Okay, see. This is the website which makes scholars. Scholar dot google dot com. Okay, scholar dot google dot com. CGKRK Meenakshi Sharma. Right. She's the wife of Dr. Tiwari. Right.
Wow.
Okay. And this is you see Rakesh Kumar Tiwari. Okay, okay, accept all of this. So you see Rakesh Kumar Tiwari,
Mhm.
you see Meenakshi Sharma,
Mhm.
you see Nagla Salem Al Sayed,
Uh-huh.
you see Hagdu, you see
Uh-huh.
Dr. CK Visparan, right?
Oh, wow.
Uh, tumor targeted delivery of siRNA using fatty ASL CGKRK peptide conjugates. Very difficult thing. Oh my god. It's very easy. CGKRK. What is this CGKRK?
Cysteine,
Uh-huh.
uh,
Glycine,
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 K, I don't remember that one.
lysine.
Lysine,
Uh, R is arginine.
arginine, and then lysine.
K is lysine, right? So
Mhm.
this is 5 peptides. So this is CGKRK. Okay. And now if you see this, very tedious. Wow, how I cannot learn it. Oh my God, I really cannot learn it. But it is very easy, right? If you
Mhm.
see this diagram here, now what is happening here? We have cysteine here. This is cysteine. Go to the structure of the cysteine. You will see the cysteine. This is glycine. This is lysine. This is arginine, and this is lysine.
Mhm.
Okay, but you cannot learn this unless you don't know the structure of the amino acid. It's very easy, and we have a very powerful tool which can make it, and I can just make it right in front of your eyes, which is called ChemDraw. ChemDraw. So you are very fortunate that we have ChemDraw.
Mhm.
Okay, I mean I'm just showing you ChemDraw. It will just take a minute to open, but I'm just saying if you are able. I mean what I told you was difficult.
Um, I think if I hadn't known, if you hadn't gone through each amino acid with me, I wouldn't have really understood. But I think it was pretty clear to me.
Now it's clear, right? It's easy. It's not difficult, right?
Yeah.
Then I have to make the amino acid. Okay, I'm writing here C. Okay, so CGK KRK,
Mhm.
right? And then I just select this and I right click here and I say here uh, expand label,
Oh,
and this is the structure here.
oh, cool.
Got it?
Got it.
So it's a cysteine, this is glycine, this is lysine, this is arginine, and this is lysine. Okay?
Mhm.
So CGK. If I ask you, my peptide, what I'm working on, I'm just deleting this all. So what you need to know that you have to install the ChemDraw on
Mhm.
your computer, okay? And
Yep.
that you can get easily from the Chapman University softwares, right?
Mhm.
I am making a peptide which is called HR. Okay?
Mhm.
Okay. So what I will do now, I will just select it and I will go to biopolymer and I will expand label. Oh, this is so drowny. This is so scary. Scary. No, it's not scary. It's just histidine, arginine, histidine, arginine, histidine, arginine, histidine, arginine. Okay.
Mhm.
So I just make it little less scary, just to explain you one more thing, which is peptide. So I have just two amino acids now, right? I will just write two amino acids, which is H and R, and I'm expanding the label here. Biopolymer expand label, yes. Now, okay. Uh, so this is here. Okay. This one thing. Uh, yeah. No, sorry. I don't want to do. I want to separate it here from here. So what is I can do? I can do here what I want to show you. H, H, and I am here writing R. Okay. I am expanding label of it. Expand the label. Okay. Now just see this structure. This is H. This is H.
Mhm.
See here. This is same. This is same. This is C double bond O,
Mhm.
and then this is OH. What? Why this is here? I don't want this here. Ah, sorry, sorry, sorry. I don't want this. I don't want this. I will just focus here. See, what is this? C double bond O OH,
Carboxylic acid.
right?
Mhm.
And what is this C?
The alpha carbon.
Yeah. And what is this?
The amino
NH2.
or
NH2.
amine.
Amino group, right? And
Mhm.
what is this? R,
Mhm.
right? So we have C, C double OH, NH2, and R,
Mhm.
right? And here we have this C,
The alpha
and we have
carbon.
this NH2, and we have this C double OH,
Mhm.
and when this, we have this R.
Mhm.
Okay. Now what I will do, I will just remove water from here. If I remove H and OH, what will happen?
Um.
So I just
So,
remove it. I just remove it.
so
I
it's
just remove it. I just remove it. Got it? And
Mhm.
now I make a line from here to here. So this new bond which is formed C double O N H. This is peptide bond.
Mhm.
You got my point? No, it's difficult. Please be with me. You have not to worry. You don't have to rush. Okay, I can start with this is difficult structure. I can start it with a simpler structure first. I can start with G and G, which is very easy. G and then I have G. Oh, sorry. Uh, G. Oh, sorry. I will just expand label, and I will expand label. So unless you appreciate this, how its structure is formed and how is it written, you will not be able to understand it. So this is how it. This is C, and with this C is H, and this is COOH, and this is NH2.
Mhm.
And again, this is C, this is COOH, and this is NH2. Got it? So what I will do, I will just remove this OH, and I will, I think there are two structures here. Anyways, so I will remove this, I will remove this, I will remove this, and I will just connect these two with a line. So this C double bond ONH bond is called peptide bond, right? So basically what happened is we removed water and COOH just connected with NH2, and that becomes CONH. CONH. So I will say CONH. If you can see this here, maybe in my notes that I showed you, I can go down and I can show you in detail more of it. Yeah, this is what I am saying. This is amino group, carboxylic acid group, and this, and I can show you here. Yeah, so I don't know. I didn't make the structure. So basically, you can do yourself like how peptide bond is formed. So basically, you just have amino group, and you connect them. So basically, if you can see here in this case as well, let me show you. Let me show you. Yeah, this is structure. This is glucose, and this is glucose. We have this H and this is OH. We just combine them. We just remove water from here, and now we have this thing connecting with this thing. Okay, so you just say see H O H, because here previously it was OH, this one, and previously here it was OH, but now this H is gone, so we just have O, so O is here. You got my point?
Mhm.
So basically this is how it is. So if you see here, we have OH here, we have OH here, and we have H here, we have H here. So if I connect this will become HOH. Yes, HOH. Got it? So, but if you go detail, if you go into this, you will realize this is not difficult. So we have this group and this group. Let me try again to, I mean, unless you appreciate this, unless you understand this, it's very difficult for you to move forward so I will start with alanine. Alanine is little easy. Sorry, Control C, A, because it has CH3 group. A, and then we have, sorry, again A. Yeah, so we have these two A. I'm expanding the label. Expanding the label. Biopolymer, expand label. You see this C double bond O NH. Previously, this NH was NH2,
Mhm.
and this C double O was C double bond OOH,
Mhm.
right? And now from OH was removed and H was removed from here, and that is the reason this connected with this. And so this new bond which is formed is called peptide bond. Not clear, not clear. Clear or not clear?
Clear.
No, little bit not clear. Focus. Okay, let me focus. Let me focus it more. G, and here I will say G. I will expand labels here, and I will ask you to differentiate these two structures and see what is this. Okay, and I have this. What is the difference between these two structures?
So,
Oh, this is G. I have to make A. Sorry.
okay.
I have to make A. and I have to okay, sorry, expand label, expand label. Now,
So when you drop the OH and then
we just compare this. This is
the
same. This is same.
yep,
This is same.
yeah,
This is same.
yes.
What is left from here?
So
OH is gone.
mhm,
OH is gone. And this is same.
mhm,
This is same. The only difference is that it's flipped. It is just flipped. It's just flipped down,
mhm,
right? It is just flipped down. If you see this C double bond O is upper side, this is on the lower side, this hash is on the upper side, then this is NH.
mhm,
This H is removed, right? And
mhm,
this H is removed, and we have NH, and this is NH.
mhm.
Make sense?
Yes,
Sure?
yes, positive.
You sure?
Yeah,
Which new bond is formed in this structure?
the peptide
Which
bond,
one? This one or this one?
that one that you just highlighted right now.
Yeah, this is. Then this is the new bond which is formed, right?
Mhm,
So you need to know that this structure is actually flipped. I have to flip it.
mhm.
Okay, I have to flip it. Flip, 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 flip. Now it makes more sense.
Except with the stereochemistry, you have to
Yeah,
change on that bond.
I mean this is here. This is like dotted, and this is black.
Uh-huh.
This is same, right? NH
Mhm,
is same. H is gone,
got it.
and OH is gone here.
Mhm.
And if we can combine OH with H, what will become?
H2O.
H2O. So H2O is removed, and this new bond is formed.
Mhm.
That is what we do. So you need to understand this. So I was going back here, which was saying this structure, this one, this is the same thing. CGK. Okay. So we have the CGK RK. So wherever you have CONH, it is peptide bond. CONH. This is
Okay,
CONH. 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 Now here I will tell you just one thing. There is two bonds. What is amide bond and other is peptide bond. Okay. So write a sentence. Peptide bond is always an amide bond.
got it.
While amide bond is not necessarily to be a peptide bond. What is the difference?
Peptide bonds are always
Amide.
amide bonds, but amide bonds don't always necessarily have to be peptide bonds.
So if the bond is formed between carboxylic acid group and amino group, carboxylic acid group and amino group, if the bond is between carboxylic group and amino group, that is always amide bond. Learn it. What I read, what I told you, learn it.
If the bond formed between carboxylic acid and amino acid groups
Not amino acid group, amino group.
or amino group, it is always going to be an amide bond.
But if the carboxylic acid and amino group are present in amino acids, that is peptide bond, right? What I mean to say is that carboxylic group can be anywhere. Amino group can be anywhere.
So they don't need to, for this one, they don't need to be alpha to each other. Is that what you mean?
No, what I mean to say is that uh, if the carboxylic acid group and amino group belong to an amino acid, then this is peptide bond.
Okay.
But if carboxylic acid group and amino group, they are not related to amino acids, then this will be called a amide bond.
Got it. Okay.
What did you get?
So if the carboxylic acid and amino acid are both present in the amino acid,
Mm-hmm.
or amino group are both present in the amino acid, then it's a peptide bond. But if the bond formed between the carboxyl group and amino group,
Mm-hmm.
if the bond is formed between those groups, then it's always going to be an amide bond.
Right? So basically, although this is an amide bond, but we call it peptide because this is present between two amino acids. Mm-hmm. Right? Uh, but if it is not the case, a carboxylic acid group can be from another thing, and amino group can be from another thing, not necessarily amino acid. But that bond still be formed, but that will be called amide bond, not uh, peptide bond. So peptide bond is exclusively for amino acids. Okay. What is the assignment for you today now?
Would it be just to go over everything and really understand?
First and second thing is that I'm going to send you a few things in your email. Review thing, for example, that 20 amino acid group, and I will ask you to read this paper, right? I will send you this paper, and there will be a lot of things, but it is very easy paper. It appears very, very difficult, but it is not. Believe
Okay.
me, it is not. It is not. It is not. It is not. This is all high school.
Okay.
Okay, another thing I want you to know for on your own, considering the same pathway, same way. I mean, I didn't teach you anything. I just teach you the way to see things in science, right? How do you see things? Okay, so you have to tell me what is ribonucleic acid, which is RNA, and what is deoxyribonucleic acid, which is DNA. Okay, what is gene? Okay, what is transcription? What is translation? I think there you can find little here in these notes that I will send you. Uh, no, you didn't find it. But I mean, whatever you come up with, I don't mind it. I will explain you. But I just want you to. I mean, I think in our high school these things are taught. DNA, RNA, transcription, translation. They are taught in our high school. What do you think? They are taught or not?
I don't think they're taught well at all, but
Okay,
I think
not well. It's another question. Not well is another question. But they are taught.
yeah, very
That is,
basically.
yeah, they're basic. So they're very basic things, right? But we don't know it. So we can come up with I don't know what is your schedule, what is your time availability. So I mean, I will be available next Sunday this time for you, and I will build up on. And this is my experiment, in fact, because I have been doing this all with all the people, but I didn't record it. I just want to record it for the greater good of the next students.
Okay, got
All
it.
right. and
Mhm.
I wish your permission for recording purpose, right? So that I can share this thing with other people, right?
Sure.
I'm
Yeah.
not sure. I'm not sure whether, because I will first share it with you,
Mhm.
but then I will just, I mean, if with your permission, obviously, right? Because I'm very much touchy about the confidentiality of the things, right? So I hope you learned something today. Science is easy.
Yeah.
What do you think?
It could be if you are willing to make it easy.
Okay. For now, do you think it is, and you will realize the impact of these little things after 3 to 4 months.
Mhm.
Okay.
Definitely.
Yeah.
Mhm.
So with this is from me now. I told you a few questions to ponder,
Mhm.
and I asked from you. Next time I will come. You come up with me, and I will say, Sarah, want to make the structure of QRWCGLK, and you will just pronounce the names of that.
Okay.
Got it? And
Mhm.
then I will ask you, what is amino acid? You will say it's not difficult. It's very easy. It's amino group and carboxylic acid group connected to central carbon, and it has a side chain which is R, which is different in 20 amino acids. Okay, are the proteinogenic amino acids are different from the other amino acids? Yes. Proteinogenic amino acids are always connected with the central alpha carbon, but there are many amino acids in which carboxylic group and amino group are present, but they are not connected with the central alpha carbon. If they're not connected with the central alpha carbon, they are called unnatural amino acids or non-standard amino acids. Right. Then I will ask you, okay, uh, how the amino acids are combined to each other. You will say, oh, condensation. What is the opposite of condensation? It is hydrolysis. Can you give me the example of hydrolysis? Yes, digestion. What we eat a lot of stuff and all is broken down into smaller pieces. That is called digestion or hydrolysis. Right?
Mhm.
Okay.
Okay.
See you next Sunday
Okay.
with
Sounds
some
good.
more science.
All right. Thank you so much for meeting
And
me.
I'm sending you these notes. Give the feedback to Dr. Tiwari. And once this sequence is recorded, I will send you, okay? On the
Okay.
Google Drive, obviously, right? If you
Okay.
need help from it. Okay. Have
Thank
fun
you.
in your life. Bye-bye.
Thank you. Bye.
Bye.